Welcome to this introduction on regenerative medicine. My name is Ian McNeice and I work at the University of Texas MD Anderson Cancer Center, which is in Houston, Texas. The objective of this lecture is to introduce you to the field of regenerative medicine and some of the diseases that may be treated and possibly cured using regenerative approaches. We'll discuss the types of products needed to improve tissue function or heal tissue defects. Each year, many patients die due to failing organs and tissues. The clinical need for tissues and organ replacements rapidly increases each year. Approximately 150,000 individuals are hospitalized each year with organ or tissue deficiencies in the United States. Of these, 10,000 die annually. The estimated cost of these transplantations is about $400 billion annually in the US alone. A number of areas that are applicable for tissue and organ transplantation include skin, neural deficiencies, cartilage, blood vessels for the heart and large small vessels, liver, pancreas, intestines, and blood transfusions. Regenerative medicine really consists of multiple types of approaches. One is to regenerate tissue. This would be in a situation where we can identify the cues that allow for regeneration without scarring. A perfect example would be the ability to grow a new limb. We would need to understand the local cues or signals that stimulate stem cells to actually differentiate and start to organize within structures and develop the new limb. Repair, which involves stimulating the tissue at a cellular or molecular level so that the tissue can repair itself. Then we have replacement. This is a biological substitute which can be created in the lab that can be implanted to replace the tissue or organ of interest. Cellular-based therapies represent a major approach in regenerative medicine. Cellular products are aimed at certain diseases where damaged tissues need to be replaced. Uses mostly only cells and very little other materials. Number of diseases include type 1 diabetes, where we wish to transplant new islet cells, adult stem cells for heart disease following such occurrences as myocardial infarctions, neuronal transplants for Parkinson's disease, bone marrow transplant for various blood cancers, and muscular dystrophy and polio. Tissue engineering is another approach that's used in regenerative medicine. It typically involves the use of scaffolds and requires an optimal environment for cell growth. Current methodologies have been developed and we can create tissues such as skin, bone, cartilage, intestine. There are presently applications for skin replacement that are in current clinical use, and the utility of using scaffolds and tissue engineering will expand in the near future. Scaffolds consist of various textures and materials. They are structured in such a way that it encourages cells to grow. They may be impregnated with other molecules such as growth factors to stimulate the proliferation and differentiation of stem cells. And they allow nutrients to permeate such that they can reach the cells within the inner structures of the scaffolds. They need to be biocompatible and biodegradable so they will not harm the patient when they are implanted. For more complex organs, such as the liver, heart, lung, and kidney, these are complex systems which involve multiple cell types. To reproduce more complex organs, we'll need to be able to have scaffolds that can integrate the multiple cell types They'll need to have metabolic functions that are needed for the function of the organ, such as electrophysiology with the heart, having electrical signals that stimulate the muscle cells to beat. So there will be a complex of both physiological and cellular events that need to be carefully orchestrated in such a way that the function of the organ is normal. A couple of examples of the types of regenerative medicine that are being explored and actually in practice today. One such example is the development of new bladders. In this system, cells are harvested from the patient. They are seated onto a scaffold and then grown in the lab for several weeks. This results in a structure that has the appropriate cell types. And so then that structure can then be implanted into the patient and attached to the bladder, in which it can then integrate. Bone repair. There have been approaches where bone marrow cells have been harvested from patients. They are seated onto scaffolds, and then the scaffold is implanted into a different area of the body than the bone structure will be needed. An example would be reconstructing a jaw from a scaffold and implanting that under the armpit, where the cells can get the signals that they require. The bone can then be harvested and then implanted in its appropriate position. In this setting, we're using the body as an incubator, as it contains all the appropriate favourable factors that are needed to really stimulate the development of the stem cells or the seeded cells that have been integrated into the scaffold. Polio is another disease. This occurs when viruses attack the cells in the spinal cord. In this situation, the signal no longer reaches the muscles in the leg, and so muscle wasting occurs. Stem cell treatment is being explored, and the goal is to encourage new spinal neurons to grow and help new muscle to grow. For diabetes, there are approaches looking at microcapsules where islet cells will be integrated into these microcapsules. The microcapsules can then be implanted in appropriate places within the body, and the microcapsules will protect the islet cells from autoimmune effects, which have been the cause of the diabetes in the first place. In this situation, the islet cells can then produce insulin, which would be secreted and resolve the diabetes. Bone grafts are another area where scaffolds can be molded to different shapes and sizes. The advantage of this is you can model directly upon replacement areas that are required. For example, with fractures, you can establish scaffolds, seed them with mesenchymal stem cells which will form osteoblasts, and in this setting you then recapitulate a bone structure. The scaffold can't be too big or the cells inside will die as they are starved for oxygen. And so these are structures that need to be carefully designed, the stem cells carefully integrated, and then appropriately cultured to generate the appropriate bone structures. Artificial skin for ulcers and burns has been generated with a product called Appligraph. This is another example of combining a scaffold, in this case with skin cells, with fibroblasts, keratinocytes, and endothelial cells, and in this situation you can have a skin substitute. An area in surgery which is of great need is the generation of vessels. Many surgeries require the harvesting of vessels from other areas of the body, again using scaffolds which can be molded into appropriate vessel shapes. Different various lumen sizes can be developed, various lengths of vessels can be developed, And so again, these scaffolds are molded. They can then be seeded with stem cells and possibly fibroblasts, endothelial cells, and cultured until we have an appropriate functioning vessel that can then be implanted during surgery. Examples would be for bypass surgery, where a vessel is essential for the success of the operation. So in summary, regenerative medicine offers new hope to patients with previously untreated diseases. There are many challenges to producing and delivering the optimal treatments and producing the optimal cellular products, incorporating these cellular products into the appropriate scaffolds, and generating more complex tissues down the road, which will be a major feat to be undertaken. And once we get to these situations, then we need to conduct carefully designed clinical trials, which will be needed to demonstrate the efficacy of these cellular therapies. Thank you for your attention, and I look forward to discussing more aspects of regenerative medicine in the future. Thank you.